Bismillahirrahmanirrahim. Hello everybody. This is Fatemeh Salabi. I work in Laboratory of Scorpion Research from Razi Vaccine and Serum Research Institute of Iran. Today I would like to talk about one of my projects. The topic is about the isoform identification and characterization of scorpion venom hyaluronidase. Among the most dangerous scorpion found in Iran are Hemiascorpius leptorus, that is from Hemiascorpionidae family, and Untitled Chapter, that is from Butidae family, can be mentioned. The venom hyaluronidase is one of the important toxic components contributing to local and systemic symptoms such as severe and fatal anaphylactic reactions following envenomation. Hyaluronidase is acid-active enzyme, and glycosidase belong to the glycoside hydrolase family, which due to its catalytic role in the polymerization of large hyaluronic acid molecules into low molecular weight fragments, play a critical role in systemic envenoming. The tissue damage by venom toxins lead to increase of abundance of bioactivity of hyaluronidase, thereby accumulation of hyaluronic acid, which in turn stimulates the immune system and produces an inflammatory response. In addition, hyaluronidase has been shown to increase tissue permeability to fluid during envenomation and to act as a spreading factor of venom. In the current study, we identified the hyaluronidase sequences of venom gland of the scorpion species of Hemiascorpius leptorus and Untitled Chapter using next generation Idomina sequencing technology. For this purpose, firstly, the scorpion samples were collected and total RNAs were extracted from the tailsome of the samples, and that was used for cDNA library generation by Idomina HiSeq platform. After preparation of the cDNA libraries, the raw data were trimmed and quality assessed by means of Trimomatic and FastQC program. After that, the qualified rays were de novo assembled by use of Trinity software. After that, transdecoder pipeline was also used to predict the open reading frames and retrieve the base untranslated region and coding sequence regions. The next step is identification of multiple isoform of hyaluronidase. For this, searches for homologous of hyaluronidase against the NCBI, SwissProt, and PFAM databases was done using BLASX and BLASP. After that, to further homology searches and remove the unrelated sequences to glycoside hydrolase family, the local scorpion hyaluronidase databases was also generated using the manually reviewed venom proteins and toxins of UniProt databases. And after that, the amino acid sequencings which representing hyaluronidase annotation were extracted from mRNA candidates, which that indicating the possible presence of hyaluronidase isoforms. The next step was investigation of alternative splicing events. For that, the intron-exon map of the hyaluronidase gene was identified based on exon coverage of RNA-seq data with the reference genome. In this study, we use one of the scorpion whole genome shotgun sequencings that are available in the NCBI as a reference genome. The transcript abundance stimulation was also done in terms of FPKM by RSEM software. After that, the structural homology analysis also was done. For that, the amino acid sequences of hyaluronidase from two mentioned scorpion species were input to the Swiss model online software. Then the quality of the predicted structures was evaluated via ProCheck through the SAVES version 6 and also by Z-score through the ProSA web software. Finally, the UCSF camera software was used to structural alignment and visualization of the predicted structure. After that, for phylogenetic analysis, we use this study hyaluronidase sequences from Androctonus crassicoda and Hemiascorpius leptorus, plus the other previously reported sequences of scorpion toxins, Apis mellifera and spider that specified for hyaluronidase. All of these sequences were further aligned using the cluster W alignment tool, and the multiple alignment obtained were used to stimulate the phylogenetic tree with MEGA-X with the neighborhood joining method. This is one of the first reports of the presence of hyaluronidase isoform in Hemiascorpius leptorus and Androctonus crassicoda venoms. Furthermore, we developed the filtering method to identify the exon intron pattern and alternative slicing events of hyaluronidase in scorpion species. Our findings show that the scorpion hyaluronidase gene contains 5 exons and 4 introns, as you can see in the picture, and also it undergo 2 alternative slicing events. This picture shows the alternative slicing events of scorpion hyaluronidase. A transcript denoted as ACH1 is related to Androctonus crassicoda, and in this study we identified one transcript with the 3 isoforms for Androctonus crassicoda, but in Hemiascorpius leptorus we identified 3 transcripts as denoted as HLH1, 2 and 3. And for each of them we identified more than 2 isoforms. The results show that this exon skipping and alternative slice site altered the expression of the isoform, and as shown in the picture, the full-length sequencings have the highest expression level, as you can see in the picture. A comparative modeling analysis of full-length isoform was also performed, in which the alpha helix, B sheet, number of extension chain, and the section were similar between 2 species. Moreover, in the present study, we have used the simulated phylogenetic tree to infer the origin of these proteins from real data in the different species of scorpions, spiders, and bees. Our phylogenetic tree suggested that the Buthidae family had a completely different evolutionary process from other scorpion families. while the Hemiascorpionidae family is similar to other scorpion species, as you can see in this picture. Given other results obtained from the analysis of hyaluronidase sequences, it can be concluded that this study methods resulted to detection of exon intron pattern and also alternative slicing events of hyaluronidase. In general, using this strategy confirmed the presence of sliced isoforms. Furthermore, by using this study method, we can evaluate the relationship between different species of arthropods. The protein sequence data reported in this paper appear in the NCBI as a following accession number, as you can see in this slide. Finally, I would like to thank Razi Vaccine and Serum Research Institute of Iran for their financial support, and I would like to thank all of you for your attention, and I wish all of you have a good time and see you soon. Bye.